So thank you, Greg, and thank you, Joao, for the opportunity to share our work with you on gene-modified immunotherapies. This is Bill on the right and Emily on the left. Bill was our first patient treated with chimeric antigen receptor T cells in August 2010. Emily was treated in April 2012. Now, how many of you have been personally touched by cancer? Maybe someone in your family, a dear friend, someone else that you know. Bill could be your father, your uncle, your brother. Emily could be your daughter, your sister, or your niece. I'm going to tell you how Bill and Emily are still here. They both had advanced forms of leukemia, had exhausted all other options. But first, I want to tell you about the problem in cancer therapy. And the problem is that cancer is our cells. It is our own cells gone awry. So how do we target something that is our own cells? And in particular, thinking about cancer immunotherapy, your immune system evolved to distinguish self from non-self. So how do you tell cancer then? And that is problem number two. The cancer-specific immune cells, if they're present at all, are very rare. And it's not only like looking for a needle in a haystack, it's a dull needle because of the tricks that cancer uses to hide from and evade the immune system. So how do we then design T lymphocytes to hunt cancer? And the answer, I'm going to take you back and use a historical analogy, is the chimera. So many of you know that the chimera is a mythical beast with the body of a lion, the head of a goat, and the tail of a serpent. In our case, we are using a molecular chimera. And that molecular chimera is an antibody on the outside of the cell and ways to signal T cells on the inside, T cell signaling domains. And so we combine the chimera with the Trojan horse. This is the first known depiction of the Trojan horse. And we need a Trojan horse because we need a way to carry the genetic material encoding that chimeric antigen receptor, or CAR. We need a way to carry that inside the cell. And so we use HIV. It's a disabled form of HIV whose only job is to carry double-stranded RNA inside the cell that is reverse transcribed into DNA and permanently integrated into the cell. And so in cartoon form, here's how that looks. There's the T cell up top, has its native T cell receptor, and the lentivirus HIV comes and delivers that genetic material that's incorporated into the genome, and that allows the expression of that chimeric antigen receptor on the surface of the T cell. These T cells are now bispecific. They still retain their endogenous T cell receptor, but now they're redirected to find an antigen on a cancer cell. And when that T cell engages the cancer cell, it can kill it. Now that's the cartoon version. And so next I'm going to show you a short 8-second movie of what actually happens. The T cells are clear. The tumor cells, we've colored them green here. And what you're going to see is this T cell engage a tumor cell, release enzymes, puncture holes in the membrane of a tumor cell, and that tumor cell is going to explode. And you can see that explode. Now, this T cell will now go on to kill the cell next to it. It's going to divide, create daughter cells, still with chimeric antigen receptor, and go on and kill more tumor cells and more tumor cells and more tumor cells. Now here's Bill in August 2010. He had relapsed several times with chronic lymphoid leukemia. He had no options. On the top, you can see before his chimeric antigen receptor T cell treatment, or CAR T cell treatment, all the brown is leukemia. His bone marrow was riddled with leukemia. After CAR T cells, we reported in August 2011 he was clear, and I'm pleased to say that almost 8 years later, he is free of leukemia and leading a healthy life. Now, we treated 3 patients in August and September 2010. Each of those patients had kilograms of leukemia. In parentheses, between 1.3 and 3.5 kilograms of leukemia was killed in each of these patients by their own engineered T cells. And here's a picture last October of Bill. You can see he's looking well, and he has a T-shirt that we gave out at the Cancer Center after this therapy was approved. You can read the quote, "The clouds went away and there was no leukemia," and he's holding a sign in his right hand. So can anyone think what he might write on that sign? It says, "I was patient number 1 of cohort 19 and all I got was the T-shirt and remission." So he's driving around the country in his RV, enjoying life and his grandchildren and his dog. Now, Bill had chronic lymphoid leukemia. The question next was, could those T cells outrace acute lymphoid leukemia, which is a very rapidly advancing disease. And these are really patients that are falling off the edge of the world and doing so very quickly. And so here's Emily in April 2012. She was diagnosed in May 2010, had relapsed twice, came to Children's Hospital of Philadelphia and received high-dose chemotherapy that would kill an adult, and only 1/3 of children make it through. She did make it through, but her bone marrow was 70% leukemic blast, and she had lesions in her kidney, liver, and spleen, and she was not far from her demise. So she received her engineered T cells on this day in 2012, and this is last year. Next month, actually May, will mark 6 years, and she's doing well. And her parents have started a foundation. They have become patient advocates for this type of therapy. They go to benefits. They meet famous people. So Emily was invited to the White House for the announcement of the President's Precision Medicine Initiative. And Tom, her father, got a call a couple days before this, and on his cell phone, the woman said, "This is the White House, and the President would like you to come down for this announcement, but you cannot tell anyone where you're going." So they get to the White House, and they're ushered into a room before the event to meet the President, and he comes in. And Tom, Emily's father, shakes his hand and said, "Mr. President, I'd like you to meet my daughter. She received her own engineered T cells and she had leukemia." And the President says, "Stop, Tom. I know all about Emily." And so he got down on one knee to shake Emily's hand and said, "Emily, how are you doing today? Is there anything I can do for you?" And Emily said in a quiet voice, "Well, none of my teachers know that I'm here." I really could use a note for school. So he got out his pad. And I'm told the school accepted this. She might have used it more than once because he didn't date it. But so Emily was the first patient, but certainly not the last. And in taking this therapy from Philadelphia to global, from boutique to global, we established an alliance with Novartis to conduct a worldwide clinical trial. And this was a 25-center, 11-country, 4-continent trial. So imagine an autologous product. Each patient has their cells collected, shipped to New Jersey, where the Novartis manufacturing center was. The gene is inserted, the cells are grown up, they're tested, they're cryopreserved, they're shipped back to each patient. If you had asked me 3 or 4 years ago, could this be done, I would have been quite skeptical, but it was done and it was performed successfully. and the results mirrored what we saw in the single-center trial at Children's Hospital of Philadelphia. And just to remind you, these are patients with limited to no conventional treatment options. The median overall survival is 3 to 9 months. The treatments that are available are highly toxic. And the conclusions from the pivotal trial were that there was high efficacy, durable, complete remissions. The Philadelphia data was confirmed in a global setting with manageable short-term toxicity. Now think about this drug for a moment. It's a gene-modified cell. It's a living drug. It's dividing. So how do you dose a drug like this? How do you think about this? It's a different paradigm. So Novartis submitted the data from this pivotal clinical trial in early February 2017, just last year, a little bit more than a year ago. And in July, the FDA convened a meeting of outside experts to provide advice to them. This was the Oncologic Drug Advisory Committee meeting. And the drug that we developed that we called CAR-19 was tisagenlecleucel. So I'll invite you to say that 10 times to yourself in your head. And at this advisory meeting, this was a public meeting. Novartis presented, Steve Grupp from Children's Hospital, the FDA presented, and then there were patient families, parents that spoke to the committee, recounting their experience. And Don O'Connor spoke, Don McMahon spoke of his son, Connor, whose dream when he was 3 years old was to play hockey. But 60 days after this picture was taken, Connor was facing a diagnosis of acute lymphoid leukemia, and 100% of his bone marrow was packed with leukemic blasts. And Connor's dad, Don, said to the committee, as you're thinking about voting on recommending this therapy to the FDA, think about this. In the 12 years since his son has been diagnosed, 6 of them were spent getting chemotherapy, 8 hospitalizations, 136 appointments at the cancer center, 4 surgeries, 23 bone marrow aspirations, 40 spinal taps, and thousands of pills taken. His treatment on the clinical trial for CAR T cells, 3 months, 4 days of chemo, 3 hospitalizations, less than 20 doctor appointments, 2 surgeries, 3 bone marrow aspirations, 3 spinal taps, and under 200 pills. So which would you choose if this were your child? You have a chance to make history by voting to recommend approval to the FDA. So what do you think they did? The vote was electronic. The chair said to the committee members, punch in your vote, and it was unanimous to recommend to the FDA approval. Now, this was July. The approval date, the date by which the FDA could recommend or deny, was October. We didn't know what day it was, but we were very optimistic based on this vote. So we were waiting from July until potentially October. I was in August giving a talk in Boston in the afternoon, and in the morning checking my email, and one comes in from Novartis with the subject line, congratulations. And it was FDA approval, the first FDA-approved gene therapy. And to mark this occasion, the Cancer Center had printed these T-shirts. They didn't know what day, but on that day there was a flash mob of hundreds colleagues, maybe 1,000 in the lobby of the Cancer Center. So imagine being amongst this crowd of people celebrating this historic first FDA approval, and you are in Boston, sitting on the Massachusetts Turnpike, having to enjoy it vicariously. And I never thought as a PhD I would see something that I was working on developed into an FDA-approved drug, let alone the first gene therapy. But here it is. It's a bag of cells. but it is a drug. And we are now in the era of synthetic biology where we can reprogram cells. We can create logic-gated Boolean cars and or not cars, safety switches, conditional cars, armored cars, and we can reprogram cells in all of these diseases, leukemia, lymphoma, myeloma, and in solid cancers where it's more challenging, glioblastoma, pancreatic, ovarian, mesothelioma, prostate, breast, and melanoma, and we have work in HIV. And on the other side of the coin in autoimmunity, we're working on regulatory T cells to suppress an aberrant immune response, as you see in diabetes, graft versus host disease, and other autoimmune diseases. So we are here. This is a car assembly line. It's a very manual process, and each comes off the assembly line quite slowly. And it took several decades for Detroit and other automobile manufacturers to get to a setting of automation, and that's where we need to go. But we really need to go here, where we have Teslas and Google self-driving cars that act automatically and persist long term. I'd like to close by thinking about the structure of a scientific revolution, and I realize I'm in London. I apologize for showing George Washington, but in the beginning, people say you're a little crazy, and you have to have a long time horizon. The next phase, they'll say, well, it may be possible, but maybe not worth doing. And through teamwork, the last phase is they'll say, I knew it was a good idea all along. And I'll just wind up by saying that this is a very complex drug. In the near term, there will be outscaling. In the mid to long-term automation, there are allocation ethics. There is financial complexity to an expensive drug, but for a one-time single infusion has long-time durable benefit. And we must think about the next generation of new therapies investing in basic research. So I will stop there, but recognize the patients that have volunteered for these clinical trials. It is only through them that we are able to develop new medicine. So I'll stop here and thank you for your attention.